Now, we set some homework last night, and we asked you to find out for your country or your territory what natural enemies have been released against weeds. So in this training, we are focusing on weeds. We're not focusing on insect pests and using natural enemies to control insect pests. We are focusing on the weeds. So can you please type in which country you're from and what you have found out, like how many natural enemies have been released against weeds in your country or your territory. So we'll just give you a minute. Somebody could report back from each country. French Polynesia, 3. And feel free to type in what they are as well, if you wish to. Thank you, Samoa. American Samoa, we don't have a number. RMI 1. So Michael can be checking to see if they've got the numbers, got the homework right.
Yeah, I'm doing that now.
Tuvalu, none, that is correct. I know that one's correct. So who else haven't we heard from? Haven't heard from Vanuatu.
We've got French Polynesia.
Guam, 2.
French Polynesia is right.
Solomon Islands, Lantana and Giant Mimosa.
Guam's a little bit under. They're missing a few. It's more than 2. So I'll just check Solomon's.
Sorry, this is just for the weeds that I know of. I know a bunch of others for the insects.
Great. What we find when we do this exercise is people are often surprised by how many things have been released in their country. They often aren't aware of them. And that's because the work was often done maybe in the 80s or the 90s or even earlier. And because the weeds are often well controlled and they slowly disappear and shrink on the landscape, people forget about them. And unless the stories are told, unless the information is being shared, then people don't know that these things were ever released. So there's actually a lot more that has been released in the Pacific to control weeds for natural enemies than most people are aware of. And Michael shared that paper and also the Excel that you can look up. And we are hoping to make this information a lot more accessible soon on the web, where it'll be very easy to go and look up for any country what's been released and what remains as an opportunity. So moving on, did you also find out for the natural enemies that have been released, whether there is more work needed to maybe spread them around? For those of you who have multiple islands and the weed is on many islands, do you actually know if those natural enemies are everywhere that they are needed? Or is there more work to do? Okay, so the don't knows are winning the contest at the moment.
And that's not surprising, really.
Exactly, that's not surprising for me either, because we are aware that not many of you have the resources to go and survey and look on all your outer islands. And so my feeling, and Michael would agree, that there's probably quite a lot of information we still need to gather. We know things have been released, but we don't know if they're everywhere they need to be or if there's more work to be done to spread them around. So yeah, the don't knows are definitely in the lead. So that's a piece of work for the future. to turn those don't knows into knows.
Lily, I got a question for you regarding that, whether the yes or no. My question is, you know, like you presented, like what was presented yesterday. The biocontrols work really well because it's a slow progress over time. And what we do have is, as was mentioned, people are impatient. So even though the biocontrol is working and the biocontrol is out there, people are impatient and they go and they grab it from where it's working and, you know, force feeding it into these areas that they want control immediately. So in that case, we can say yes, because people are just impatient and can't wait for it.
I think if people are really keen to get involved, that's actually a really good thing. I haven't heard that story from too many countries, so that's wonderful if people are really, really keen to get involved. So we're going to move on.
Sorry, look, I'll just take up that in Papua New Guinea and also places in other countries that I've worked in the Pacific, people, the local villagers will see something happening and they'll move insects around. The gallfly on Cromalina was moved around by many villagers in some of the other provinces. So it is something that we encourage because we can't always get to every island, or we can't get to every village or every area. So we actually encourage that sort of stuff. But I think the question that Lindy was asking was whether there's scope for more movement. And I think in most countries, the answer is yes. Obviously, some countries don't know if there is or not because they don't know what's out there. But I think in most countries in the Pacific, there is scope to move agents around. And that's really the crux of the question there.
Thanks, Michael. So this next slide, I just want to share the vision that we have with you. We want to foster and create a strong network of people throughout the Pacific who are confidently using NENS, natural enemies, natural solutions, that are sharing their lessons, their stories, sharing natural enemies with each other and expertise. And by doing this, we're allowing and empowering you to reduce the harmful impacts of invasive weeds. So obviously, to get from where we are now to this vision, there's quite a bit of work to do. And we started with the photo here was from a course that was held in Apia back in 2019. And we had a great group from all over the Pacific who came, some of them for 5 weeks, and learned about all sorts of invasive species work, including the NENS. And we just need to keep building on this, and through the Pacific Invasives Learning Network, through online sessions like this, through information sharing, we're setting up, Timo's helped me set up a Facebook page. We can communicate, and we just need to put a bit more thought into how we're going to link you guys all up. It was amazing yesterday to see the map and dropping the pins in the map where you all are. If we can all, even though we're so far apart, be together like this and sharing, we can deliver on this vision and make a big difference for invasive weeds. So what we're going to do now is, yesterday we talked about the philosophy, and now we're going to move on to the practice. How do you actually do it, and what are the steps that are involved? So today we're going to talk about the first 2 steps, and then tomorrow we're going to talk about the next 2 steps, and then the next day we'll talk about the last 2 steps. It's quite a lot of information to cover, so we're just breaking it down into bite-sized pieces. I also just wanted to say where we are currently working with NENS programs. So at the moment we have funding and we are working in these places. There is question marks at the moment beside Samoa and Fiji, because while we have the funding and the mandate, we actually still need to have conversations with people in those countries about whether they want to be involved. So if you're sitting somewhere else and your name is not on that list, but you are interested in getting involved in NENS, please contact myself. My email address is on the front of the PowerPoint, or Timo, and talk to us. We will have to find a way to get you involved, and that might involve us having to apply for funding. But if we know you're interested, we can find a way to include you as well. So please get in touch if you are interested and would like to be involved in NENS. So when you do a NENS program for weeds, we always follow the same steps, and we're going to work our way through these steps and explain them. But also we do have this available as a poster. In fact, it's up on the Battler lounge there on the wall. And if anybody would like a copy of the poster, we can share it electronically, or possibly even get some printed and post for you if you would like to have one for your wall. So I'm just going to hand over now to Michael for a minute. We did mention yesterday that this isn't something that we've just made up. This is international best practice. So Michael, would you like to just explain what that looks like?
Okay, thanks, Lindy. Morning, everyone. So as Lindy explained, or we explained yesterday, that weed biological control has been occurring for over 100 years. And we've been evolving of how we do this practice. And because all these countries are involved and we're moving agents around the planet, there's been a series of best practice guides put up. So in the top one there, Joe Balcy-Ernest put up a paper in 2000, and FAO have had produced 2 code of conducts or guidelines. And the FAO 2017 guidelines is now available for sharing. So while we'll refer to it, you can get it off the Google Drive. It's a 16-page document, and it just goes through guidelines on the export or the importation of agents, how they're treated, how we release them, how we test them, all these things. So, and a lot of this course, this training follows these guidelines. So next one, please, Lindy. So I'm just going to run through a few of these. We've tried to summarize those guidelines into 12 dot points, although I did find another one we could add. So what we want to do is we want to ensure that the target weed's potential impact justifies the release. So we're not looking at backyard garden weeds, which you can pull out by hand. We're looking at weeds that cause an impact to agriculture or biodiversity on a large scale to justify the cost and the time involved in releasing natural enemies. Most importantly, we want to make sure that we've got multi-agents approval for each country. So more often than not, it involves agriculture departments or ministries and environment departments and ministries. And this is to ensure that everything is done above board and the right procedures and protocols are taken place. So for some countries who have done biocontrol many times, there's set procedures in place. Other countries, they may not have these procedures, and they're not hard to develop. We want to select agents which have potential to control the weed. So we're not going to waste our time introducing an agent which is not causing any impact on a weed in any other country. So if we look at Lantana as an example, there's 20 biocontrol agents in Australia, but not all of these would be recommended for release. It's not because they're not safe or they're not so specific, but they're just not doing the job that we would like them to do. So we only want to look at importing agents which have got a known track record of controlling the weed in at least one other country. More importantly, we want to make sure that our agents are host specific. We talked a little bit about that yesterday, that we don't want to have our agents attacking other plants. And they've also got to be approved agents. They've got to be bona fide and tried in other places. And we want to ensure that our intended agent is released. So what I mean by that, we don't want to release things which is where there's a contaminant in our population. And the point which is missing, and I think it pretty needs to be put in here, is when we're importing biocontrol agents into a country, we usually import them into a quarantine or a secure facility. And this is because sometimes insects are collected from the field of one country and brought in and sent over to another country. And so when they're collecting them in the field, you might find there's a mite on there, or there's a thrips or something on the leaves as well. And so if the shipment goes into a secured facility, you can then look at what's in that shipment, and you can pick out only the insect that you want, and you can destroy or autoclave or burn the rest. In many times, if the insects are coming from Australia or New Zealand, they're going to come from a laboratory culture, and we've already done that process for you. But insects have moved around. The cider beetle was moved from PNG to Fiji, and from Fiji into Vanuatu. And so a lot of that material was field collected. So going into a secured facility is really important to make sure that you're not introducing inadvertently contaminants. We also want to make sure that we use the appropriate protocols for release and documentation. And what this means that when we're releasing our insects, we release them in the adequate numbers to get establishment, or we release them in the appropriate places, or even the timing. So if we're releasing a seed feeding beetle, we want to make sure our plants are seeding at the right stage for those insects. If we've got a leaf feeding insect, we want to make sure our plant is in full leaf for our insects, and then the right numbers. And there's some protocols and some guidelines on how to do that. It's always good to document things. Lynn, you mentioned that yesterday, that if we keep documents, we can work out whether why things aren't working. We can go back through our records. We also want to monitor the impact on the target, and there's a couple of reasons for here, and point 8 refers to that. First of all, we want to find out if our agent has established, because if the insect has established, we don't need to keep putting our insects at that particular site. We can move on to other sites. If the insect is feeding and it's been there for a year or two and it's not been very effective, we don't want to keep putting more and more insects out, because that might be just a waste of resources, and might be better to look for another agent or natural enemy. And also, obviously, if the weed becomes controlled, we also don't need to put out any more natural enemies, because the weed is already under control. So monitoring is really important, because if you've already put insects out at one site and they're well established, you can then go and choose another site or another island. It's a better use of resources. Now, point number 9, we did talk about whether agents or natural enemies may attack other species. And while we can be pretty confident that the agents that we supply to the Pacific and other countries are host specific and unlikely to attack anything else, one of the guidelines has been that we should look at non-targets anyway. And this is one of the requirements for Australia, is that when releasing a biocontrol agent, we do check other plants, just as a matter of course. And not surprisingly, we don't see any of non-targets, but we need to check just to keep our regulators happy and peace of mind. We also want to encourage the assessment of changes in plants or animal communities. And this could be in the natural environment or even in agricultural systems, where we can find that farmers are increasing their yields because the weed is less. And we did that work in Papua New Guinea when we were working on Cromalina, where we were able to document changes in yield by farmers in their crops and plantations. We'd also like to see or monitor the interaction amongst the natural enemies. And we do this for a few reasons. Sometimes when you put out multiple agents, they work well together, which is great. That's what we'd like to see. But occasionally you might find that one agent is antagonistic to another. And we just need to be a bit wary of that, because that might affect or direct how we direct future work. So we need to look at that. And I think it's also important to communicate our results to the public. They need to be aware of what we do along the way. This is part of our initiation steps. But we need to embrace and let them know that not only have we put out an agent so they can monitor it or see for themselves, but we can also share our wins. And so that if a farmer is spraying his weeds and there's a biocontrol agent out there and it's very effective, then maybe we can, the farmer can save some money and time by not spraying his weeds because it's going to get controlled by the natural enemy. And just keeps everybody in the loop. And then the farmer can utilises his resources in another way. Next slide.
It's you done. Thanks, Mike.
Yep, I think that's it.
Yeah, so the take-home message here is that we are sticklers for best practice. If you're getting involved in NENS, we want best practice to be adhered to, and we encourage you to look at the international best practice guidelines and download them and read them. Now, thank you to Joseph. He's just pointed out that this post is actually available also on the shared Google Drive, so you can access it straight away after this session if you're interested. So I'm going to talk to you now about exploring feasibility. So it's a kind of a look before you leap. The first step is we've got to look at whether or not is this project going to be feasible? Is it going to be difficult? What are we up against? So we need to ask ourselves questions like, what is the desired outcome? If the desired outcome is rapid eradication, then we know this is not going to be appropriate for NENS. You might need to look at other techniques such as herbicides. But if it's a widespread weed that's well beyond eradication and you have the time and the resources to go down the NENS approach, then that would be a tick in the box for proceeding. We have to ask ourselves, the question is, well, are there acceptable other control methods? I mean, if there are, then it might be more important to look at NENS for another target for which there is no acceptable control. Some weeds are very difficult to control, as I'm sure lots of you will know. Some of them are really hard to kill, even with herbicides. So we ask ourselves the question, well, why might natural enemies be needed? And it might just be that there's just so much of the weed and it's so expensive and it's so extensive that we know this is sort of our only hope and our last hope often. We have to ask ourselves the question, what is the risk of actually not exploring natural enemies? And I touched on this yesterday. If we don't do something about invasive weeds, we know there's going to be a lot of harm that is done. Those weeds are going to affect livelihoods, the ability for people to grow food, to access resources they need. They can affect human health, animal health, fresh water, not even mentioning biodiversity or ecosystem services. We can't afford to do nothing about weeds. Doing nothing is not an option. So what we have to do is think about what is the safest, most practical and most sensible option. We need to understand how the weed is related to other plants. And this is very important because, as we mentioned yesterday, and we'll go into more detail tomorrow, when we're working with highly specialist natural enemies, the plants that they're most likely to be able to attack are the plants that are most closely related to the target weed, because they share the same chemistry. So we need to be able to work out for your countries, what plants do you have in your countries? So what is related to the weed, whether it's a native plant or an economically important plant or just maybe an ornamental. We need to draw up a list of all of the things that are related. And if there are, native plants, that's obviously it's super important, and economically important species we don't want to harm. That's also very critical to know. If it's just an ornamental plant that maybe a few people have got in their garden, it might not be a make or break situation if we're deciding about whether to proceed with natural enemies. But we really need to have all this information at our fingertips. We also need to know, are there any known natural enemies that could be used? So has anybody done this work anywhere in the world? Are there agents already in the Pacific? And then there's a whole lot of stuff we have to do. These end up being quite thick reports, as you can imagine. I said yesterday that often there is somebody who likes the weed, who uses it for something. So is there going to be somebody or a group who is upset about the potential for controlling the weed? We need to understand, are people using the plant for medicinal purposes or for making fence posts or for floral arrangements? We need to have all that information. We need to know, are there going to be other obstacles? We know, for example, that some countries at the moment, it is very difficult to get natural enemies, to get the permits to export them. We know with COVID at the moment, it's quite hard to move things around the globe. We need to understand what's going to get in the way of maybe doing this project. We need to be able to comment on what is the likelihood of success. And in session 5, Quentin, my colleague, is going to touch on this a bit more. He's done quite a lot of work looking at how you predict which projects are likely to be successful. And we know where a project has been successful in one Pacific country, that's often an extra big tick that it's likely to be successful in another Pacific country. We need to figure out, well, what work needs to be done? Do we need to do testing? Who can do the testing? Where will it be? Where will we source the agent from? How long is this going to take? What permits do we need? How much is all this going to cost? Have we got funding? And then yesterday we showed some pictures from the Cook Islands where we had 3 invasive vines all growing together. And we knew there we actually needed to tackle all of those 3 together. Otherwise, we're going to end up taking out one and it will just be replaced by the other 2 and we'll be no further ahead. And another really key question here too is this weed the top priority? And again, we're going to talk a bit more in the session 5 about how we actually determine priorities, because I think, again, I said this yesterday, the problem with weeds is there's so many. We're not talking about 1 or 2, we're not probably even talking about tens, but you've probably got hundreds of weeds that actually need to be tackled. And we've got to figure out very carefully what the top priorities are for each country. So an example of we did a feasibility study quite some time ago now for the Peltate Morning Glory. It's quite often called Maremia or big leaf rope. It's got the great big leaves on it and a very robust invasive vine. And when we asked and looked at the information to see whether or not it's actually introduced or native in the Pacific, we got all this really conflicting information. And so you will see on this map here, some people said it was native, some people, so the ends, the green ends, some said it was introduced, the red eyes, unknown, the yellow U's, and A was absent. Now, if this plant is actually native to the Pacific, then it's possibly not an appropriate target for using for natural enemies. So we've had to try and untangle this complex situation, and we've been doing some genetic work to try and understand, has this plant been in the Pacific for a very long time and is native, or does it only have a subset of the genetic material you might expect and therefore it's more recent introduction, or is there more than one form? Maybe there is a native form and an introduced form. We haven't completed this work because we haven't been able to get enough samples from all the parts of the Pacific that we need samples from. And once COVID prevents travel, it's going to be a priority for us to get around and ask you guys to help as well to get more samples so we can resolve this question about, is this an appropriate target for the Pacific or not? And then that information can be discussed at some regional workshop yet to be determined. So as I explained, it's not unusual for people to have some uses for weeds. And you know, African tulip, it's got pretty flowers, it provides shade. I've seen fence posts made out of it and growing into new trees. The mylaminate is often used, I believe, for wounds and is very effective. So where we have the situation of conflicts, we need to weigh up very carefully the beneficial uses against the harmful impacts. And sometimes that would need to be an economic analysis to help, which might say that the beneficial uses are only worth a very small amount of money and the cost to the country is many, many thousands or millions of dollars or whatever currency. And then overwhelmingly, therefore, it is more important to control it than to keep it widely available for the beneficial uses. But it's really important that, you know, communities are involved in these decisions. So you don't get a long way down the track with a project to find that your community is outraged by the suggestion that you're going to control a plant that they like. And there needs to be that conversation with the community about the harm those weeds are causing and why there might be a necessity to go down the natural enemies track. And if the community is really still opposed, it might not be a good idea to proceed. However, because as we mentioned yesterday, that the natural enemies will never eradicate the weeds, usually there is still some middle ground here where people can use them. They will still be there. There will always be mylaminate leaves available in any country where the mylaminate rust has been released, because the vine will always be there, just less of it, but there will be enough leaves if people want to find them. It's also possible sometimes for us to take a different approach where, often this is possible with if you have an invasive tree. If you like the tree, you can just stop the tree from making so many other baby trees by attacking the reproductive structures, by finding natural enemies that would attack maybe the flowers, the pods, the seeds. So you can have your tree, but it doesn't make thousands more trees every year. And that has worked very well, often as a compromise. But I must, you know, reiterate, this is a conversation that communities need to have and needs to be resolved very carefully before you embark, so you don't run into trouble. So now I'm going to ask you again to get your phones out and to give us some feedback using Mentimeter. I'd like to hear from you about what you think might make it easy for you to use natural enemies in your country or territory. So if you're not quite sure, things that might make it easy would be things like having supportive community or good legislation, track record in using natural enemies, good champions. Tell us what might make it easy, and then we'll get on to the hard stuff. So, oh yes, here we go. Success stories, simple permitting process, funding, facilities, education, buy-in, willingness of community, policies, champions. Good. So it's great that people can identify some things that is going to make it easy for them to do NENS, but we always have to consider the flip side. What's going to make it difficult? And I know some of you have got some challenges here. What are the difficulties that we're going to have to overcome if you don't have a NENS program? Or maybe you're just starting out on one and you think there might be some challenges. Tell us using Mentimeter. Lack of funding, it's always a good one. Biosecurity legislation. Convincing your government and your communities, your politics. Limited knowledge. Extreme weather patterns, that's an interesting one. We might talk about that when we talk about releasing an establishment, Michael. Land issues, inaccessible areas. So we can see there are plenty of challenges here as well that we need to be mindful of, and we will need to troubleshoot. And that's again where I think it's really important that we act as a whole Pacific network, because each of you will be able to give advice to another country about how you've overcome some of those challenges. And for me sitting here in Auckland and for Michael sitting in Brisbane, we don't always know all the answers, and you will have answers that you can share. So that's why I think you're going to be able to teach each other a lot. We can teach you some things, but you're going to be able to teach each other a lot. And that's why I'm very passionate about this idea of a Pacific-wide supportive network.
Can I just make a quick point about this? Convincing government came up quite high, and this is not unusual for many countries, not only in the Pacific, but Asia and Africa, where countries which haven't done biocontrol, they're a little bit reticent about it. And this is where these training workshops and also this sharing of success stories may just help countries which are a little bit reticent to have buy-in. And that's, if we can work through that and share all our knowledge, then some of these countries which haven't done biocontrol or are a bit reluctant, even those ones which haven't done it for 20 years, they may just start seeing the benefits of it. So, let's just keep sharing the stories and see if we can get people's buy-in.
I think another point there too is seeing is believing. And if we are able to organise for more visits between countries, people can come and see what other countries have done and see with their own eyes and hear the stories from the people. That might help as well. So that's just another tool we can have in our toolbox potentially if we have some resistance. So yes, please, now with Mentimeter, what are the sort of conflicts of interest that what are some of the problems, some of the risks we're going to have to address? So we've got obviously quite a few people have supported medicinal invasives. Can you give, if you can, give us some specific examples of weeds that you think people have uses for, that they might feel conflicted about the weed being controlled? So if you're able to put specific examples of weeds in, like the African tulip, for example. Does anybody use the Maremia for anything useful? weeds used for food. I know in New Zealand, our beekeepers like quite a few of our weeds here to feed pollen for their bees. And yes, and sometimes there are native birds that will be feeding and using some weeds. We have in New Zealand a very giant insect called a weta, which is about as big as my hand. And they only survived in a particularly prickly weed with lots of thorns on it because the rats weren't able to get to them there. And so we had to resolve that conflict, that actually what we needed was better rat control. So these very special, iconic insects could survive, but we could control the weed. Lantana used for honey. Okay. So, yeah, the whole point of the feasibility is really making sure we understand completely what we're up against, what our options are, what we're going to need to do, and if we're ready to actually step in and do the project, or whether we need to back off and get more information or get more buy-in. So I'm going to hand over to Michael now. He's going to talk about the next step. If we've decided to proceed with the project and we've got the funding, what do we do next?
Thanks, Lynley. So when we're looking at, or we've nominated our target weed, in many cases we know where the weed is found. So it's things like water hyacinth, we know it's found in South America. But occasionally we strike a weed where it's cosmopolitan, or it's found in a number of areas, or it's got a wide distribution. And so often we need to do some molecular studies to work out what exactly that weed is and where is the best place to start searching for natural enemies. So if you look at a weed like lantana, lantana is found from southern USA all the way down to Argentina. So that's a fairly large area to start surveying. But with molecular studies, we can actually work out where did lantana come from in Australia, and we can target those areas that, like the center of origin, if you like, where we're going to get the best match between the weed in Australia and the weed in South America. And so molecular studies are able to do that. And we're doing this now with 3 weeds that we're targeting in Vanuatu: Pico or Solanum torben, wild peanut or Senatora, and Urina lobata or hibiscus burr. And so we collect leaves, and then we can do some genetic from a number of sources, and we can do some genetic analysis. When we're also doing the surveys in the country, we want to find out if there's anything already there. So occasionally, if there's a weed present in a country and there is a closely related native to that weed or in the country as well, it might actually have some natural enemies already on it. And we've seen in Vanuatu some insects attacking our 3 target weeds, but obviously they're not doing much damage because the weeds are still quite present. But it's worth looking at what's already out there. And there's another reason for doing that. If the weed is has been worked on before, an agent may actually already be there, spread from another country. We want to gain that information about to help identify natural enemies that may exist to complement. So some of the things that we look for is if we do find native insects in a particular country and they've got some parasites on them or some predators, are these going to attack our natural enemies that we introduce? And sometimes if we introduce a insect which has got a close related insect on it already in the country, the parasite might move across, and that may impact on how well we can do biological control or how well our natural enemies can work. So it's good to do these surveys. We also want to get some baseline data, and I'll go on to the next slide for that one.
Yeah, just before we move on to the next slide, this weed in the background here is the one Michael referred to as Pico or Solanum torvum. And we have in our containment facility here in Auckland a very exciting beetle that we're in the final stages of testing, and we think is going to be a very useful natural enemy for the Pacific. So if you have a problem with this Pico, that's some good news for you for the future. Also, I'll just point out this bag here with the orange stuff in it. This is silica gel. And so when we do the molecular studies, we just get a small leaf and we put it in the silica gel, and it's the same stuff if you buy a camera, or sometimes if you even buy some vitamins, they have the silica gel that just dries things out. And then that's sealed up in a bag, and then that can be sent to a lab such as the one we have here to do the analysis. So it's actually quite simple to collect the samples. A bit harder to actually extract the DNA and get the data, but the sample collecting is not difficult. Oh, we seem to have lost the baseline data slide. It's okay.
Okay, so
Is it after this one? Okay, sorry, looks like we've gone, it's out of order. So here we go. We'll jump forward and then we'll leap back.
okay, so the in baseline data, what we mean by that is some data to start off with, so we can actually monitor whether our biocontrol agent is making an impact. And so some of the things that we want to do is what weeds are present, and usually by this stage, we're actually after our particular weed. We want to find out whereabouts it is. And there's two reasons for this. This is, we get an idea of its distribution, but also when we're putting our natural enemy out, we want to make sure that our natural enemy is released in all areas where the weed is present, or all islands. We also want to look at how much of the land is infested. So are paddocks 100% infested, 50% infested? And again, this allows us, when we put out our natural enemy, to work out whether or not our agent has made a difference to those infestations. And the harmful impacts of the weed, what we mean by that is the impacts maybe on biodiversity. So we might look at other vegetation, especially the ground covers, to see how much is there. And then if the weed disappears, we can see if that the ground cover and the other species increase. But we can also do this for agriculture, for cropping. We can see how much yield is being produced, or productivity of land by farmers, how much grazing area is occupied by the weed. And then when we've released our biocontrol agent, we can find out if the impacts have decreased and productivity increases. Some of the other things which is always nice to do, and we don't always get a good indication of this, is the cost of current control. So if a farmer or someone is spending $100 per week or $100 per month or whatever, then is that cost of control that the farmer is spending to control that weed, is that decreasing after we put our natural enemy out? And this is really useful because we're able to document the cost-benefit ratios or the savings to the economy by our natural enemies. The other thing Lenny just talked about was the beneficial uses of that weed. And we can monitor whether or not, if we decrease that weed, are the beneficial uses going to be impacted? And if we're looking at things like Mycania, where we only need a few leaves to treat cuts, then maybe there's not much differences in the beneficial uses. Things like the trees or other species, then we can look at, was there going to be an impact on that? And ever since I've started working on biocontrol, there's always been this push for photos. And this is partly because 20 years down the track, we forget how bad a weed was. And so what we want to do is take before and after photos, and there's a session on that in monitoring. I think that's session 5, could be 4, where we look at how we monitor the impact of our biocontrol agents. So if we take photos, and I think there's a couple of photos I mentioned in our presentation yesterday, before and after releasing natural enemies on water lettuce, on water hyacinth, where we had lakes covered in the dam, covered in the weeds, and then 4 to 5 years later, those waterways are virtually clear of the weed. So photos are really great because they remind us of how bad something really was, but it shows you the impact of our natural enemies. And we'll talk about, as I said, we'll talk more about that a little bit later. Should we go into the next slide?
So I think what we're going to do is
We've got to go back.
We're going to do a Mentimeter first, Michael,
No, we missed
and
the
ask
slide.
about the baseline data, then we'll go back to the overseas surveys. It's got to slide out of order here, so this is the next logical one to do. So
Okay.
as Michael has explained, we really need the baseline data about weeds if we're going to be able to tell our stories and measure changes in the future. What I would like you to tell us now, on a scale of 0 to 5, have you got no data about the weeds in your country? Or have you got really comprehensive data? You know what weeds you've got, you know where they are, you know how much they cost, what impacts they're having. Can you score, please, where you sit on that scale? Okay, so we're hovering just around 2. Any further refinement of that? That doesn't surprise me. I know even in New Zealand, we don't have the kind of baseline data we would like. And I think there's definitely some work for all of us to do to try and get better baseline data. So we've landed there on around 2.3. So if you answered yes, you have got baseline data, can you tell us what you've got? Have you got photos showing weed infestations? Have you got maps, GIS? So you can say how many hectares or acres of a weed you've got. Have you got reports? Tell us what you've got.
It'd be also nice to know if they've found on other islands how widespread it is, especially in countries where there's many islands. Do people know which islands are impacted?
One of the really good things about everybody having a mobile phone, they've often got a pretty good camera on them. And so it's never been easier to take photos. And it's very cheap. It's we're going to talk about more about this when we talk about assessment, but we all need to get snap happy and take as many photos as we can, because photos, especially ones from decades ago, are so amazing to be able to show what's changed. And people will sometimes say, oh, it's the same as it always was, and you get out a photo and you show the difference and it's not the same. Okay, so we do have some reports and some maps and some GIS and aerial photos. That's really good too. Great. Thanks everyone for that. If you don't have any baseline data, and if you don't, you won't be on your own, because it's often something there is very little of. What do you think are the barriers that we need to overcome so we can collect it? Is it funding? Is it trained people? What is it? The lack of computers, lack of cameras, too much work to do, other priorities? Okay, so funding is not surprisingly coming up as an issue. Training, just how to collect it, how to store it, having people that are trained. Specialized equipment. Fortunately, you don't tend to need too much specialized equipment. But yeah, having the people. Turnover in staff. Yes, you train up people and then they move on to another job.
And quite often it's a matter of people not knowing what sort of data to collect. And I think we talk about that a little bit later on, but
We will.
often people don't realise how simple or how easy some data can be easily collected, such as photos or a GPS point. So it's not surprising that people haven't collected data because often they don't know what to do, or they think it might be quite complex when in actual fact it doesn't need to be.
And thank you for whoever pointed out about iNaturalist. So I'm not sure how many of you have ever looked up iNaturalist, but it's a way where people can report observations of biodiversity, and people post pictures of things they don't know what they are, and experts will have a look at the pictures and provide advice about what they think the identity is, and you can create maps and all kinds of things. So if you've never had a look at iNaturalist, maybe after this session you might want to Google and have a look and just see what information is in there for your country, but you can use that as a way to tap into experts too to help you to identify things. So thanks for pointing that one out. Right, I think I now need to jump back to the other slide. It's going to let me go back. Yes. Whoop. Gone too far. There we go, Michael. Now here you go.
Okay.
So we've done the native range. Sorry, we've looked in country, in your countries, at what's the situation. Now Michael's going to talk about the overseas part.
So once we've got a bit of an idea of what we've got in our own country, we can then start looking at native range surveys. And this is particularly for those weeds that no work has been done already. We're not looking at either new targets or novel targets where there's no natural enemies already in use somewhere. So if the natural enemies are not well known, we need to conduct surveys in the native range. And this is where the previous slide we were talking about, trying to do the molecular studies and things like that, working out where we're going to survey. So we then start our surveys, and once we've identified our country, we can then start our surveys. So the photo here is a group of us working in Malaysia, looking for natural enemies on Senatora or wild peanut. This is on behalf of the project in Vanuatu. And we're working with a local organisation of scientists there. And so we start surveying the plants, looking for whatever we can find on those weeds. And then we can, once we've got the insects or the pathogens back in the lab, we can then start to prioritise. But also during these surveys, we can look to see whether the insect is doing any damage. And we need to be a little bit careful about insects that may be just resting on the plant, or are they actually causing some sort of impact, chewing the leaves, boring the stems, feeding on the flowers or the fruits. We also want to then study the life cycles, and we can do this in the lab or in the glasshouse situations. So we can actually see how long it takes from egg to adult, number of stages. We can get a bit more idea of the kind of impacts that insect may have on the plant. We can grow plants with and without the insect to see how the insect, what sort of damage it does, whether it stops flowering or fruiting or things like that. And then we can develop some rearing methods. And this is obviously very critical because eventually when we need to host test the insect, we can design our host testing along with its life cycle. And so we need to understand our insect before we can start testing it. And we're going to talk about testing tomorrow, so I won't go into any more of that. So I'll try to get as much information from the native range as possible. And then we're able to then prioritise those 1 or 2 agents that are what we think is the best ones to bring back for further study.
So I might just add in here, we talked yesterday about the cost. So when we are developing natural enemies for completely new novel targets, this is a lot of work. You can imagine having to go and do overseas surveys and find out what natural enemies might even be available to work with. And sometimes we're finding things that are new to science, and we have to be able to find people who can even identify them for us. And we might find maybe as many as 100 different natural enemies on the weed. So there's quite a lot of work to figure out from that 100 which ones might be the top 5 to study, and then work out how we're actually going to work with them and grow them on so we can do the testing. So this is quite an expensive part of the project, and it can take years. So I just wanted to provide that additional bit of information. So I'll just go back to now where we need to be, and I think we're going to have another Mentimeter session. Right, yes we are. So, okay, if there's any questions that you've got that you'd like to ask us about the material that we've covered today, please submit them. And again, remember that you can like the questions that other people are submitting, and we will answer any questions that you have. Nothing's popped up on the screen yet. Hopefully you're typing in long and involved questions to keep Michael on his toes. Okay, so the first question has come in. Has a conflict of interest ever been an issue during your work? And can you give us an example? So Michael, I might hand that one to you.
Yeah, there has been. Occasionally it comes up. In Australia in the 1980s, we were going to look at, well, not my organisation, but Australia was looking at controlling the pasture weed. And it was a similar example to the one that Lindy used, where the weed was used as a foraging plant for bees to make honey. And because this weed was widespread, it was an easy source for bees. However, the weed itself was still used as a biocontrol agent because it affected pastures and it was toxic to horses. And the argument was that the impacts of the weeds were far greater than the benefits of the weed, and that there are other plants that the bee farmers could use other than this weed. But it was a very interesting exercise to start looking at how we deal with this conflict of interest. And in fact, it was the start of what they've got now in Australia as the Biological Control Act. And it's an act which we can start using, or how to help sort out conflicts of interest. In general, many of the times there are no conflicts of interest, or the conflict of interest is minor and can be easily handled. But there was a case Lindy mentioned in about using plants for timber or shade, and in South Africa they started importing agents that only attack the flowers or the fruits or the seeds, so that people can still use the tree, but it doesn't escape plantations and invade waterways and natural ecosystems. So it does come up from time to time, but we just need to make sure that everyone's had their say, and any conflicts of interest are aired, and people have got you know, a right of comment.
I know the participants from Hawaii will well remember the conflict of interest over the strawberry guava that delayed that project for some time resolving. We have managed to resolve quite a few in New Zealand, the beekeepers, the medicinal herbalists, by being in good communication. There is one project we haven't proceeded with here, and that's against a conifer, which is a really bad tree weed here, but it is of concern to our production forestry sector, that if we bring in natural enemies, there may be problems for the forestry sector. So we haven't proceeded with that one at this time. So you have to understand what you're up against, and sometimes make a call that maybe you shouldn't proceed and you should work on something else. So thank you for that question. We'll move on. What are some issues that might come about during rearing? Michael, do you want to start with that one?
I actually gave a whole talk on how to rear biocontrol agents and or natural enemies. And there's a lot of issues that come out. I think the very first one is to make sure that your environmental conditions are right. We don't want the rooms to be too hot or too cold. Humidity is important. But some of the key ones is to make sure our plant health is adequate. We don't want our plants to be infested with aphids or mites or mealybugs, because that impacts on our insects. Some things that people don't quite pick up, it's not quite obvious, but the phenology of the plant. Have we got the stems the right way for our stem-boring insects? Or have we got the seeds or the flowers at the right stage, or the tips of the plants at the right stage for our insects as well? So there's a whole lot of issues to do with rearing that we need to cover. Wonder if it's worthwhile, if everyone agrees, I can put on a PowerPoint presentation on rearing. We can drop it into Google Drive if people are interested. But there are a lot of issues, and it's not just a matter of throwing insects into a cage and walking away and coming back a week later. There is a bit of delicate process. It's understanding your organism, understanding the life cycle, and understanding its needs. So there is a bit of effort required.
And I might just add there, even people who are experts at rearing insects or plant pathogens run into trouble with some new ones when they don't come with a recipe, when nobody has worked on them before. We have some we've just completely failed to be able to rear. And we've even had other ones where sometimes the female insects can choose to only have male offspring. And so you very quickly run out of a rearing culture if you have no females being produced. So yeah, if there is interest, we could even organise another online training about rearing. We're going to touch on it briefly in another session, but I think when we're getting into sort of the rearing insects, we will, if we're working with us on the project, we will come in country and we will help you, because it's like anything, it's a skill, and you have to learn all the things to avoid and the things to do correctly. So thank you for that question. Next one, elaborate on multi-agency approval for target code of conduct.
What we mean by that is that, and I did touch on it, but normally there's at least 2 organisations involved. Certainly in Australia, there's the Department of Agriculture, which approves of any importation, but there's also the Department of Environment. They don't want any imported natural enemy or anything for that matter attacking their biodiversity. And so often what we've asked organisations or countries which aren't familiar with biocontrol is we ask what departments or organisations need to be involved in approving a biological control or a natural enemy, just to make sure that everybody's on board. And it's just part of those discussions that we need to have in the initial stages. So each country usually has an agricultural department. They might have an environment department. They also may have a forestry department or fisheries, or and each country is set up differently. So you just might need to look at what agencies need to be involved in approving the importation and the use of natural enemies.
You don't want to find yourself at war with another department because there wasn't a good conversation. And in fact, what we promote is when decisions are being made about which weeds to target, you involve people right at the start. Bring people on the journey with you, and you'll find things go much more smoothly.
I'll give you a quick example. I won't name the country, but it was in Asia where we're trying to introduce start up a biological control program and introduce some natural enemies which have been utilised before. And each department wasn't prepared to talk to another department. And so quickly it ground to a halt. And so nothing got developed. But we also didn't quite know how many departments needed to be involved because they divide their departments up into very small agencies. So if things aren't out on the table, things can get held up. So we need to have those conversations.
Thanks. We'll move on to the next question. Can we share the information from the other countries about their natural enemies? So I'm not quite sure what this question's asking. Information about natural enemies is very readily available. So yesterday on the PowerPoint, we provided a link to an international database where you can look up for any country in the world or for any weed, what's been done. And then the resource that we've put on the shared drive that we have used, you can also search just for the Pacific about what's been done. So that information is there. If anybody is still stuck, you can contact us for some further assistance on that. But yes, there's plenty of information that can be shared. How's dealing with the communities feeling like this will be on top of the challenge list? And I think from what I've heard and what I've seen, that it's different for each country. Some places the communities are really happy and on board, and other places the communities are a bit more suspicious or maybe have slightly different priorities. So I think this is, if we're looking at embarking on a NENS program for any country, we need to assess that and what needs to be done. And what I always promote is that we need to start talking to the community really early, and before you're at the stage of actually wanting to release a natural enemy, there has to have been a conversation and a chance for people to talk about their worries and their concerns, to be listened to, and to come up with ways forward. That has to be done. There's lots of pre-work. There's lots goes into a NENS project. Examples of cost-benefit analyses conducted in the Pacific. I'm not sure, Mike, are you aware of anyone that's been done in the Pacific? I mean, I know ones that have been done in New Zealand.
We did it wasn't a cost-benefit analysis per se, but what we did in Papua New Guinea was we looked at the cost of, we did two studies. One was on Mycania or Malaminate, where we looked at the impacts and benefits of Mycania in Papua New Guinea before we started that project. And then what we did, and we did a retrospective study on biological control of Chromalina, also in Papua New Guinea, where we looked at what the costs were to landholders, but we looked at the benefits of that successful biological control program. But in both cases, the amount of benefits each of those two weeds had was quite small. Even with Mycania and the medicinal medicine, it didn't come out very strongly at all. But they were the only two that I know of clearly. But certainly it has been done in other countries where, especially where we want to work out whether there is a conflict of interest, we do these studies.
There is actually a lady who's sitting in the room behind me out the door. Her name's Susie Greenhalgh, who is an economist who loves doing this kind of thing. But the work that she's done that I'm aware of is looking at whether there's a cost benefit argument just for tackling invasive species. So she has done work in places like Vanuatu, just getting information about, well, which species do you target? But this is again another whole area of expertise and conversation that we could look at exploring in the future. If there's interest from this group, we could ask Susie, for example, to run a session. And I know she has done some training in the Pacific, teaching people how to do cost benefit analyses. So we have some expertise we can draw on there from part of my organisation. Okay, do you have cost benefit analysis of natural enemies before introduction? So I think we've probably covered that one off in the previous question. Okay, so thank you for all those questions. It's so good to get them coming through. I just want to again check, are you able to follow what we're saying? Are we talking too fast, too slow, using too many big words? It's making you all go to sleep? So, yes? Can follow what we're saying? That's good. Marvelous. Okay, that's really promising.
Yeah, thank you.
Again, we're going to say, is there any feedback on the session or suggestions for the next sessions? We really appreciated the feedback that came through yesterday. It was great to hear from people that they had enjoyed it, and we're always up for improvements. You know, we're humans, we're doing this for the first time. We are really wanting to deliver the best possible training for you, so we are really open to any suggestions or improvements. And it is anonymous. All this feedback via Mentimeter is anonymous. So if you're a bit afraid of, you know, making it known it came from you, we won't know it's from you, and we will appreciate the feedback anyway. So we will just hang around for a little bit longer and let the feedback come in, but also if anybody actually has a question and they can stay a bit longer and they would like to ask Michael or Temo or the team at SPREP a question, we can hang around for a few more minutes. Otherwise, we're going to look forward to seeing you again tomorrow, and we'll have the next installment of the exciting journey of using the NENS. So thank you very much for your time and attention today, and to everybody who's put effort into making this session a success.
Lindy, can I just make a quick comment before everyone disappears? There was a couple of questions about what's available for in other countries or in the Pacific with agents. And there's, as you said, there's a lot of information out there, but people can feel free to email Lindy or myself asking for papers or specific information on weeds and things like that. So, you know, if you've got some queries after these trainings or after the next week or whenever, our emails are on the front of these talks. Feel free to email us, and we'll gladly help you out and send information your way specifically for your country. So don't be afraid to email us directly.
And we are also willing to organise more sessions in the future just for Q&A. You might go away after this training and talk to people and come up with a whole lot more questions. So we will just keep organising sessions as is useful for this group and keep the conversation going. So I'll just sign off for now for those who need to leave. Ka kite anō, mā te wā. We see you tomorrow.
Yeah. So